Hello everyone, welcome to the new session on biochemistry where we are going to deal with the Krebs cycle proper. Okay, the Krebs cycle, it is otherwise known as citric acid or tricarboxylic acid cycle. It is a cycle of aerobic reactions by which the acetyl-CoA A, the energy-rich acetyl-CoA A, it is oxidized to yield carbon dioxide, water, ATP and energized hydrogen atoms, or even we can say electrons. Okay, so here hydrogen atoms are trapped in the form of NADH, or we can say FADH2, right? And these the electrons are then being pumped into the electron transport chain, okay, which is later used for the synthesis of ATP. The Krebs cycle, it occurs in mitochondrial matrix, except for one step over here, uh, the succinic acid to succinic acid to fumaric acid over here, succinate dehydrogenase. Except for this enzyme, all the enzymes of the citric acid cycle, it is present in mitochondrial matrix. Okay, succinate dehydrogenase on the other hand, it is present on the inner mitochondrial membrane. Okay, it is a part of the integral membrane or integral membrane protein of the mitochondrial inner mitochondrial membrane. Okay, now in carbohydrate metabolism, Krebs cycle immediately follows the glycolysis, while in the lipid metabolism, we can say the citric acid cycle, it follows the beta oxidation of fatty acids. Okay, now so this is about the basic things about the TCA cycle or the tricarboxylic acid cycle. Now, when you start with the tricarboxylic acid cycle, it is actually the acetyl coenzyme A coming into play. Okay, so acetyl coenzyme A is produced from pyruvate by oxidative decarboxylation process, which is catalyzed by the enzyme pyruvate dehydrogenase complex. Okay, now the acetyl coenzyme A is combines with or condenses with oxaloacetate to produce citric acid, and that is the first step in the whole cyclic process. And the citric acid produced, so 2-carbon acetyl coenzyme A combines or condenses with 4-carbon oxaloacetic acid, resulting in the formation of 6-carbon citric acid. And the citric acid, it gets isomerized to form isocitric acid, which undergoes oxidative decarboxylation to produce alpha-ketoglutarate acid, right? And the alpha-ketoglutarate acid, it undergoes another set of oxidative decarboxylation to produce the succinic acid over here. Okay, these two steps, that is isocitric acid to alpha-ketoglutarate acid and alpha-ketoglutarate acid to succinic acid, these two steps do have a transient intermediate. Isocitrate, while it is getting undergoing oxidative decarboxylation to form alpha-ketoglutarate acid, an intermediate product of oxalosuccinic acid is produced. It is a very transient intermediate substrate which readily undergoes transformation into alpha-ketoglutarate acid. Now, isocitrate is a 6-carbon compound, while because of decarboxylation, alpha-ketoglutarate produced, it will be only 5-carbon compound. Okay, and this 5-carbon alpha-ketoglutarate, it undergoes another set of decarboxylation to produce a 4-carbon succinyl-coenzyme, right? A 4-carbon compound. And this again, succinyl-coenzyme is again a transient intermediary product, so it will produce succinic acid later. This succinic acid, it will produce oxaloacetic acid through fumaric acid and malic acid. Okay, so oxaloacetate is regenerated, while the acetyl part of the acetyl coenzyme, it is getting completely converted into carbon dioxide. So each turn of Krebs cycle actually takes up oxaloacetate and one acetyl group of acetyl coenzyme A and releases 2 carbon dioxide, one over here and another over here, 2 carbon dioxide and 8 pairs of hydrogen, right? That is, it is equivalent to 8 electron pairs. Okay, and oxaloacetate is regenerated at the end of the cycle, which can be utilized for another cycle. Okay, the next cycle, while acetyl group is completely degraded to carbon dioxide and the molecules like. Okay, so there is no oxaloacetate is every time being regenerated to be used again. Now, step by step we can see. So the first step is the condensation of acetyl-CoA enzyme A with oxaloacetic acid to produce the citric acid, right? Here what happens is the first molecule, it is a 2 carbon, you can see 1, 2. Okay, do have a CoA enzyme, and here you can see the first compound, the 2 carbon acetyl coenzyme A. It condenses with the oxaloacetate. To be specific, during this reaction, what happens is the acetyl group of acetyl coenzyme A, it condenses with the carbonyl group of oxaloacetic acid to produce a citronyl coenzyme A, which is an intermediate, which readily undergoes hydrolysis. You can see water is added, which readily undergo hydrolysis to produce citric acid and coenzyme A. I'll repeat, the acetyl group of acetyl coenzyme A will condense with the carbonyl group of oxaloacetic acid to produce a citronyl coenzyme A, which is a very transient intermediary product which undergoes hydration or which undergo hydrolysis to result in the formation of citric acid and coenzyme A. Okay, and the whole process, the condensation, it is being catalyzed by citrate synthase or citric acid synthase, or it is otherwise even referred as citrate synthetase. Okay, I hope it is visible, citrate synthase. Okay, now here you can see the coenzyme A is free, and this coenzyme A, which is being liberated over here, it can be used up during the oxidative decarboxylation of another pyruvic acid molecule, okay, to produce acetyl coenzyme A once again. So here it is getting released. Now the next is we have is a citric acid. Citric acid to the next one we have is isocitrate. It's an isomerization process, but during this it is not a single step, it is a two-step process. Okay, that is a formation of isocitrate from the citrate. So citric acid, first it undergoes isomerization and then an asymmetric dehydration. So here you can see it undergoes dehydration, water is released, right? So what happens is it will get produce cis-aconitic acid. Okay, so it loses one molecule of water, citric acid, it loses one molecule of water, results in the formation of cis-aconitic acid, and this cis-aconitic acid, it undergoes stereospecific hydration, water is added. Okay, so stereospecific hydration and results in the formation of isocitrate, and both these reactions are catalyzed by the same enzyme aconitase. Okay, so here it is actually a dehydration and later a rehydration process. That is what results in the formation of isocitrate from the citrate. Okay, now what happens to isocitrate? You can see the isocitrate, it will undergo oxidative decarboxylation. Okay, oxidative decarboxylation to produce alpha-ketoglutarate acid. Again, this is not a single step. It is a two-step process where in the first step, isocitric acid, it will undergo dehydrogenation, that is oxidation, to result in the formation of oxalosuccinic acid. Okay, here we don't have the oxalosuccinic acid, again here also it's oxalosuccinic acid, and this oxalosuccinic acid, it will later undergo decarboxylation to yield the alpha-ketoglutarate acid. Okay, so first it is a isocitrate to alpha-ketoglutarate. It is a oxidative decarboxylation process. It is a two-step process. Firstly, the isocitrate, it will undergo oxidation or dehydrogenation to produce oxalosuccinic acid, and the oxalosuccinic acid, it will later undergo decarboxylation process to result in the formation of alpha-ketoglutarate acid. Okay, so this is a two-step process. Now, the enzyme which catalyzes the reaction. Isocitrate to oxalosuccinic acid, the conversion is, or the oxidation, it is being catalyzed by isocitrate dehydrogenase. Okay, and here the hydrogen which is released from here, the isocitrate, it is received by NAD. Okay, NAD plus, and it results in the formation of NADH. You can see here NADH is being released from the step. Okay, because first step it is oxidation, that is hydrogen is released, and this hydrogen is taken up by the NAD plus, where NAD plus get reduced to form the NADH. Okay, and while the isocitrate, it will get oxidized to form the oxalosuccinic acid. The second step, so the enzyme is isocitrate dehydrogenase. Okay, isocitrate is getting dehydrogenated with the help of this enzyme. The second step is decarboxylation. The oxalosuccinic acid undergoing decarboxylation, and this particular reaction is catalyzed by oxalosuccinate decarboxylase. I hope the name you can very well relate with, because carbon dioxide is getting released, isn't it? A decarboxylation process, and decarboxylation takes place. What is the substrate? Oxalosuccinate. Okay, so it is oxalosuccinate decarboxylase enzyme. It catalyzes the decarboxylation of oxalosuccinic acid to alpha-ketoglutarate acid. I hope that part is clear. The next step is alpha-ketoglutarate acid to succinic acid. Again, this is also a two-step process. This is yet another what you call oxidative decarboxylation process. Okay, so here the first step is oxidative decarboxylation process. Here oxidation is taking place as well as decarboxylation process is taking place. Okay, now the alpha-ketoglutarate acid is a 5 carbon compound, and it undergoes oxidative decarboxylation to produce 4 carbon Succinyl-CoA enzyme. Here, this particular reaction, it is catalyzed by alpha-ketoglutarate dehydrogenase complex. Okay, it is not a single enzyme, it is a complex enzyme. And here, alpha-ketoglutarate acid, carbon dioxide is released. At the same time, it undergoes oxidation. The hydrogen released is taken by NAD plus, which is get reduced to produce NADH. This is the second NADH molecule being produced in the citric acid cycle. Okay, so the oxidation of alpha-ketoglutarate acid, it is again irreversible. You can see here, it is irreversible. All these were reversible, right? These two are irreversible reactions. Okay, and the NADH, it is being released. One molecule of NADH is produced. Now what happens to the next step? Succinyl-CoA enzyme A to the succinic acid. Succinyl-CoA enzyme A is a highly unstable but energy rich compound, and this what happens is, at this particular stage, that is succinyl-CoA enzyme A to succinic acid, it is a substrate level phosphorylation step. Okay, it is not ATP dependent, but it is GTP is the molecule which is being produced. But again, it is a substrate level phosphorylation. And here what happens is, succinyl-CoA enzyme A, it reacts with GDP and inorganic phosphate to produce GTP and succinic acid. I hope it is clear. Okay, succinyl-CoA enzyme A reacts with GDP and inorganic phosphate to produce GTP and succinic acid. It is a substrate level phosphorylation process. Okay, now this particular reaction is catalyzed by succinyl-CoA enzyme A synthetase, or it is also known as succinic acid thiokinase. Okay, I hope this one will be better. Okay, succinyl-CoA enzyme A synthetase. Now this, what happens to the GTP produced? The GTP produced, it will be utilized for the phosphorylation of ADP, and ATP will be produced. So it is actually ATP generating step itself. Okay, and this GTP dependent ATP synthesis, it is being catalyzed by the enzyme nucleoside diphosphokinase. Okay, ADP is getting converted to ATP during the process, while GTP goes back to GDP state. Okay, now next step, the succinic acid is ready. Now we will go to the next step. The succinic acid, it undergoes dehydrogenation. Okay, that is, it undergoes oxidation process. And since succinate is the substrate, the enzyme which catalyzes the dehydrogenation, it is succinate dehydrogenase. Okay, it will result in the formation of fumarate. What happens to the hydrogen? The hydrogen release, it is taken up by the FAD molecule, and FADH2 it is being produced. Okay, and this FADH2 will enter the electron transport chain along with the NADH molecules produced during the cycle. Okay, so succinic acid, it undergoes oxidation or dehydrogenation, transforms into fumaric acid, and the enzyme which catalyzes it is succinic acid dehydrogenase. Okay, and during this process FADH2 is being produced. Okay, now succinic, as already mentioned, succinic acid dehydrogenase is membrane bound enzyme present on the inner mitochondrial membrane of the eukaryotes. Okay, now what happens to fumaric acid? Fumaric acid, again, it undergoes a reversible reaction. It undergoes hydration, stereospecific hydration, results in the formation of malate. Okay, right.